solution is a mixture of two or more substances in the same phase. This is formed when a solute dissolves in a solvent. A solution can also be formed when two or more miscible liquids are mixed together. When ethanol and water are mixed together, a solution is formed. Now, how would we separate these solutions? To separate dissolved salt from water, we would simply evaporate as much water as possible, leaving behind white salt crystals. The separation technique is known as evaporation. To separate ethanol from water, we must take into account the fact that these substances have very different boiling points. Water boils at 100 degrees Celsius, whereas ethanol boils at 78 degrees Celsius. This mixture can be separated by simple distillation. The lower boiling point component, in our case ethanol, will evaporate first. The ethanol vapor condenses as it passes through the condenser, and the distilled ethanol can then be collected in the receiving flask. The above mixtures are homogeneous mixtures. This means that the components of the mixture are in the same phase, or the composition of the mixture is uniform throughout. Now, we will look at separating heterogeneous mixtures, where the components in the mixture are not in the same phase or the composition of the mixture is uneven. If we have a mixture of sulfur and iron filings, we can easily separate this mixture with a magnet. The magnet will remove all of the iron filings, leaving behind the yellow sulfur powder. We know that salt is completely soluble in water, but sand is not. How would you separate a mixture of salt and sand easily, keeping in mind the solubility of salt in water? Pause, think, and continue when you have an answer. The salt and sand mixture can be added to water. Salt will dissolve, but sand cannot. The resulting heterogeneous mixture can be filtered through a funnel lined with filter paper and washed with water to remove as much salt as possible. The substance that remains on the filter paper is known as the residue, and the substance collected in the flask is called the filtrate. For our case, sand is the residue, and the salt water is the filtrate. The salt can be obtained using evaporation and the sand left to dry.
We're going to learn about 4 methods for separating mixtures, which are evaporation, filtration, decantation, and distillation. Evaporation is a method used to separate homogeneous mixtures of a liquid and solid, for example, water and salt. The mixture is left in a dry place so that the liquid component evaporates and only the solid remains. This system is used industrially to extract salt from seawater. Filtration is a method to separate heterogeneous mixtures of a liquid and a solid, for example, the orange juice and the pulp. The mixture is poured over a strainer or on porous paper, called a filter. The solid component will remain in the filter, whereas the liquid will pass through it. Decantation is a method to separate heterogeneous mixtures of two liquids, like water and oil. The mixture is left to stand so that the denser liquid is at the bottom and the less dense at the top. Once this point is reached, one of the two is removed from the container. Decantation can also be used to separate heterogeneous mixtures of a liquid and a solid, like water and sand. And finally, distillation. This is a method to separate homogeneous mixtures of two liquids, like water and alcohol. When heating the mixture, the liquid with the lowest boiling point will evaporate first. In the case of wine, which contains water and alcohol, the alcohol will evaporate first because it boils at 78 degrees, whereas water boils at 100 degrees. This big tube here is filled with cold water. When the gaseous alcohol goes through the spiral tube which is inside, it will be cooled down and returned to its liquid state.
Today's topic, digestion. I love apples.
Even I love apples. Do you know what happens to the food inside our body?
No.
Let's get into this boy's body with the apple and see what happens to it.
Wow, we have become so small.
We are so small that we can enter this boy's body. So, let us start from mouth.
Oh, teeth are making it in small pieces and also grinding it.
Yes, saliva gets added here to this food. Food becomes a thin paste.
Teeth, with help of saliva, make a thin paste. Which way to go now?
Now there are two ways. One goes to the food pipe and other goes to the windpipe. We are going to the food pipe. There is a valve called pharynx. This food pipe is also called esophagus.
Windpipe, food pipe, pharynx. Oh, now we are entering the stomach. Get ready. There's a lot of liquid here.
Food travels from esophagus to stomach.
What is this liquid?
Oh, these are gastric juices. They make the food melt.
It is a kind of boiling here.
Yes, this is also called acid.
Gastric juice.
It is small intestine.
Oh, but food is just traveling. Where would the body take nutrients from the food?
Right question at the right time. Most of digestion happens in small intestine. Most of nutrients are absorbed by body here.
Small intestine. Most of digestion happens here. What is this green liquid?
Oh, this is bile. It is produced in liver and pancreas. It helps in digestion. This liquid comes in small intestine and helps to take nutrients from the food.
Alright. Bile, liver, pancreas. What are these small hills?
These are called villi. Nutrients enter blood from these villi.
Oh, it's a long journey.
Yes, it is 22 feet long.
Small intestine is 22 feet long.
Now we are entering large intestine.
Is it bigger than small intestine?
No, small intestine is bigger in length, but large intestine is bigger in width.
Oh, small intestine is narrow. That is why it is called small intestine.
Yes, here the water from the food is absorbed.
Large intestine, water is absorbed here. Oh, I want to go back. Now this is becoming very thick.
Yes, let's go back. Now it is food waste, which body does not need. It would go out of the body through anus.
Anus! Food waste goes out.
So here you can see whole digestive system. Now you know the whole journey of food.
Wow! It is a long journey. It starts from mouth, goes to esophagus, which is food pipe. From there, it goes to stomach. Then it enters small intestine, and from small intestine, it enters large intestine. And from there, it comes out of body through anus.
Let's see an experiment on what happens inside the stomach.
Oh, what is this?
Let me show you how gastric juices in stomach burn the food which we eat.
Why are you cutting the apple?
Because food goes into our stomach in small pieces.
What is the liquid in this jar?
Oh, it is acid, like in our stomach.
So, acid would burn all the food and give us energy.
Yes, acid in stomach burns all the food, or we can say it cooks all the food to get nutrients.
Yeah, it is taking time.
Yes, stomach takes 6-7 hours to burn all the food. I want to show you the difference between small and large intestine.
Oh, these look like intestines.
Look at this. This is very long. But its mass is small. It is called small intestine.
Yeah, it is 25 feet long.
Yes, you're right.
Oh, this with big mouth is called large intestine. It is only 5 feet long.
Yes, it is small in length but bigger in mouth size. That is why it is called large intestine.
Small intestine, 25 feet. Large intestine, 5 feet.
Let's start with some definitions. The term pathogen most often describes any microorganism that causes a disease. These can range from simple illnesses like the common cold to more complex illnesses like tuberculosis. When one of these pathogens invades our body, we call that infection. Infection occurs when microbes like bacteria and viruses that exist all around us enter our bodies and begin to multiply. Disease occurs when the cells in your body are affected negatively by infection and signs and symptoms of an illness appear.
Do you know what microorganisms are and how many types of them are there? Let's learn everything about them! There are organisms that consist of one single cell called unicellular organisms. These organisms are very small. That's why we call them microorganisms. You may also know them by other names like germs or microbes. To observe microorganisms, we need specific instruments like microscopes. Microorganisms come in many sizes and forms. In ancient times, some investigators had already questioned whether or not the diseases human beings had may be caused by organisms invisible in plain sight. This is when they started investigating microorganisms. Anton van Leeuwenhoek was the first investigator to start observing microorganisms in the 17th century. Microscopes and the investigations done with them allowed other scientists to continue learning how diseases were caused or how they could be cured. Some types of microorganisms are bacteria, viruses, and fungi. Bacteria. They are prokaryotic cells. They have no nucleus. Some bacteria are useful in food fermentation, like making yogurt or cheese. By contrast, other bacteria are harmful and may cause infections. We treat bacteria with antibiotics. They spread in different ways. Viruses. They are not living beings. To carry out the vital functions, they need a host, that is, a living being. They are much smaller than bacteria. Sometimes they can infect them and live inside them. Some viruses cause diseases like flu or chickenpox. To prevent diseases caused by viruses, vaccination is important. There isn't a vaccine for every virus. Fungi. Some fungi are microorganisms bigger than bacteria or viruses. They can be found in the air, plants, or in the water. Some, like yeasts, are used to make foods. I'm sure you've seen mold on fruit or bread. This is a type of fungi. As we have seen, microorganisms can be everywhere because they are not visible in plain sight. To prevent diseases and avoid getting infected, it is very important to have good hygiene habits.
Thank you for watching. Do not forget to attempt your quiz. See you in my next class. Bye.